Welcome all. This is Dr. Mobeen Sayed with one more episode of Long Story Short with DrBeen from the FLCCC platform. The discussion today is actually really fun, and Dr. Paul Merrick had suggested that I talk about this. This discussion is about caffeine or coffee. Coffee has caffeine as well. So this is caffeine, coffee, or caffeine separately. So caffeine or coffee inducing autophagy. That's a beautiful idea. Inducing autophagy, and this particular study that I'm going to discuss is about the autophagy in the liver cells. This study is in vitro and in vivo mice study. However, a question could arise, and that is, if this study is talking about coffee's effect or caffeine's effect on autophagy in the liver cells, what will happen in other cells? So it seems like coffee will induce autophagy in other cells as well. In some cases, it is good. In a couple of studies, I've seen that there are some tissues that may not have as good an outcome. So let's start our discussion. So starting from here, this is FLCCC or COVID-19criticalcare.com. So if you go down here on the side, you would see there is a list of videos and the other informational resources present here. At the same time, there are various protocols that I think you can take advantage of. So with this, here is the study. This study is caffeine stimulates hepatic lipid metabolism by the autophagy lysosomal pathway in mice. This study is 8 August 2013, although it was again referred in 2014, and then many articles have referred to it. Here is another study, just to kind of balance it out, that would this happen in other parts of the body as well. This is actually recent study, January 11, 2022. Caffeine prevents re-stenosis and inhibits vascular smooth muscle proliferation through the induction of autophagy. So they are talking about the cardiovascular patients where they may have stenosis of some blood vessel, and maybe it was made patent by various procedures. And now we want to make sure that it doesn't become re-stenosed, and caffeine has shown to help there. And that help is through autophagy. At the same time, blood vascular smooth muscle proliferation is the number of smooth muscle cells increasing in blood vessel walls, which can be a bad thing. And then caffeine helps with that too. And then here is another study. If you see, this is caffeine induces autophagy and apoptosis in auditory hair cells via the SGK1-HIF1A pathway. And in here they said that some auditory cells, hair cells, which are responsible to hear various frequencies of sound, they may get damaged because of autophagy, because the autophagy induced by caffeine, and they said this may be bad. So the point of showing these studies is that caffeine can actually induce autophagy, which maybe nowadays, considering the pandemic, may be good, but do talk with your doctor as well. Caffeine has many other effects too, and I'm going to stay restricted to this study, and I would not be discussing other parts of the caffeine or impacts of the caffeine. Here, there is another study, Impact of Coffee on Liver Disease, Systemic Review, beautiful area, Coffee, the Magical Bean for Liver Disease. Then various other references are present as well. So with this, let's start with my drawings. So the point of this discussion is, number one, to understand the caffeine's or coffee's effect on the non-alcoholic fatty liver disease in vitro and in mice. That is one. Secondly, the more important thing is to understand the mechanism by which caffeine induces autophagy, to understand what beautiful things are happening when caffeine is taken. The third thing that we'll discuss is the dose of caffeine, and I'll spill the beans right here. It seems like the people who are consuming 2 to 4 cups of coffee daily, this study is for them. It is for moderate to heavy coffee drinkers and not for the light ones. This is why I would say once again that if you are going to have coffee as a result of this discussion, please discuss with your doctor as well. So let's start. First of all, summary of the discussion, sort of a highlight. And this would take about 7-8 minutes. Then we'll go in the details. The highlights are this. This is an in vitro study. That means they did this study on cells. plus in vivo study. That in vivo part was in mice. During this study, what they did was they did genetic manipulations of cells to turn off various genes or turn off various genes to understand the pathways caffeine was triggering. Then they did pharmacological part of the study. That is, they shut down the pathways they suspected caffeine was taking. They brought in medicines or substances to shut down those pathways to confirm that in the absence of that pathway, caffeine was not able to do its function. Then they also did the mouse study, which is metabolomic. Now, what did they find? What did they say in the study? They said the known so far is that caffeine stimulates lipid reduction in the liver. So this is known. The question is how? What is the mechanism? And for you to clarify this, in all of our cells, we have cellular machinery to reduce lipids or to metabolize them or to degrade them, mostly called beta oxidation of lipids, for example, or to take a protein and break it down into smaller units, which are amino acids, or to break down glucose and do glycolysis. Every cell has that. The researchers' question was, does the caffeine reduce lipids in the liver through these traditional mechanisms, or does it go through autophagy or autophagy? And then you can ask this question that why were they interested in doing this? So the reason for the researchers to figure out what is the mechanism is, number one, just general curiosity to understand what is the mechanism for autophagy or caffeine-induced lipid reduction. The second thing was to understand when lipid starts or fat start accumulating in a patient who is a patient of non-alcoholic fatty liver disease, that means their regular, standard, traditional mechanisms are not correctly working. And researchers wanted to see, is caffeine going to offer a new pathway, a new way? If that is possible, that there is a new way, then could caffeine be a therapy for this? And I would actually show you this study that we are discussing is 2013 study. And researchers said that, hey, in 2013, so far there is no medicine for non-alcoholic fatty liver disease or NAFLD. And here if you see, this is nowadays NIDDK, NIH, and medicines for treatment of NAFLD, NASH. And if you see here, other than asking for weight loss, they say medicines, no medicines have been approved to treat NAFLD, either NAFL or NASH. However, researchers are studying medicines that may improve these conditions. So even till to date, there is nothing. So the researchers' point of view was that we should look at caffeine in a little more detail to understand if this could be a therapeutic pathway. Now, what did they find in this study? They found that caffeine causes autophagy lysosomal pathway to become enhanced or to become induced or triggered. What does that mean? And we'll do that in discussion today. Here, let's say inside a cell, this is a phagosome. A phagosome is, or actually I should call it autophagosome. An autophagosome is a little garbage truck within a cell that is picking up the trash within the cell and collecting it. So that is this blue little truck, let's call it. And then once it is filled, it will go near a biohazard truck. So this orange one over here is a lysosome. Lysosome is filled with various enzymes that can degrade other products. So lysosome has machinery to break down other things. And lysosomes will like to have an acidic environment. And one of the reasons that caffeine is good in this situation is that caffeine also promotes acidic environment. So anyways, in short here, phagosome or autophagosome and lysosome will fuse and they take the trash out, and this whole process can be induced by caffeine. This is what they found. Now exactly how, what did they see? What is the mechanism? We'll discuss that. In addition to this, so what did they found? That autophagy or autophagy was induced. They also found something very interesting. They found that caffeine reduced an enzyme called mTOR, and I'll explain what that is. Similarly, they saw that in hepatic liver in vitro cells, the amino acid quantities became altered. Amino acids are smaller components of proteins. So inside the cell, the amount of amino acids, they changed. Similarly, in the liver cells, in vitro, the sphingolipids, these are lipids that are bound with proteins. These lipids' volumes changed as well as a result of presence of caffeine. Now, you must ask, so what? They saw these things to be less. Here is what is important. All of these substances suppress autophagy. For example, when mTOR is present, it reduces autophagy. When amino acids and sphingolipids are present in a particular amount, they suppress autophagy because they think that they are going to be used for other building material. However, when caffeine arrives, what it does is it actively inhibits all of these mechanisms. So if you see here, this is caffeine, and caffeine is attacking the mTOR and inhibiting it. Now remember, mTOR was inhibitor of autophagy. So if you inhibit the inhibitor, then autophagy would start. Or in simple words, we can say it is necessary to suppress mTOR to allow autophagy to begin. And guess who suppresses it? Caffeine. Who else suppresses it? Intermittent fasting. What else suppresses it? Spermidine and some other substances as well. But here again, we are keeping ourselves focused to caffeine. So caffeine actively suppresses mTOR, which will result in the beginning of autophagy, induction of autophagy. So all these three would help. So that's very interesting. The takeaway from the above diagrams was caffeine not only induces autophagy by having more phagosomes and lysosomes and their function, it actually also suppresses the opposing components of autophagy. Okay, so that was the in vitro part of the study and summary of that. What did they do with in vivo? Here is what they did. They had a few mice. Some of the mice were intentionally, actually all of them, were fed diets that were going to create more fats in them, going to create more lipid accumulation in them. They were not on ketogenic diets. They were not on non-carb diets. But at the same time, they were also on the diet, these mice, which would promote lipid formation. So let's say high-fat diets, HFD. Now, one group of mice was also treated with caffeine. Other group of mice was given high-fat diet or given diets that would create increased fat levels without caffeine. And guess what happened? They observed that those mice that were given caffeine, their liver did not have the lipids accumulating and their body weight did not change. On the other hand, the other mice that had high-fat diets, so diets that would promote accumulation of lipids, but no caffeine, these mice had lipids accumulating in their liver cells, plus they generally gained weight as well. Very interesting. So what's important in summary? Hepatic liver lipid content was reduced by autophagy and not traditional mechanism of breaking down the lipids. This was what they demonstrated. This is what was not known before. So this is a huge contribution towards science to say when caffeine is given, it is the autophagy mechanism that is engaged, and here is a proof of that. So with this, we stop. We stop for the summary. Now I'm going to go in the details of the mechanisms that they observed. It is a fascinating study. So let's start. So they did many experiments. That's what researchers do to prove their point. Some highlights of that. Number one, what they did was they took liver cells or HepG2 cell line in vitro. They treated these lines, cells, with caffeine for 6 hours, 24 hours, 48 hours. And the first thing they observed was that in these cells, the genes to make LC3, and I know you're going to say, what is LC3? I'll explain that in a second. The genes to make LC3 were increased. The genes to make DGAT1, adipose TG lipase, hepatic lipase were reduced. And I know once again you're going to say, what's the difference? Why? So here is the answer. The DGAT1 or adipose triglyceride lipase or hepatic lipase are traditional enzymes to break down lipids. So imagine if a cell is given caffeine and the cell breaks down lipids more. You could be scratching your head to say, was the traditional mechanism engaged and enhanced? So here they demonstrated that no, the traditional lipolysis enzymes were not increased. On the other hand, if you are saying, okay, then how did the caffeine help break down the lipids? They said LC3 was increased. Now, LC3 is an important builder enzyme. It's actually a microtubular lipidation enzyme for the physicians here, for the nurses here, NPs, PAs, medical students, nursing students. This is an enzyme that helps lipidate the microtubules, and the result of that is it helps produce a phagosome. So caffeine is inducing the production of an autophagosome. So it is enhancing the expression of the enzyme that would help build the phagosome. You would actually see down the road, this would make you so excited. You'll see down the road that not only caffeine would help make more phagosomes, it would actually help make more lysosomes as well. So not only it creates more trash trucks to go pick up the trash, it also makes more acidic trucks to destroy that trash. So this was their first clue that caffeine is going through the autophagy pathway and not through the traditional lipid breakdown pathway. Then the next thing they saw, this was very interesting. So see over here, these are fats present in a hepatic cell. Again, why do I keep saying hepatic cells? Because the study is in the liver cell, so I don't want to generalize it. Although I showed you other studies which are proving that this happens in other cells too. But because I'm talking about this study, I want to stay loyal to this study's topic. So here, these are lipids or fats sitting in a hepatic cell. And why are they saying we hate coffee? Because caffeine is going to build phagosomes, and not only build them near the fats. These phagosomes are built and then co-located next to the fats. Of course, I'm going to hate them because they know this truck is going to come near me. It's going to pick up some of the fats from here, lipids, and break them down with the help of lysosomes. So what do we know so far in the detail? Caffeine does use the autophagy. It does help make the phagosomes. It actually increases the formation or production of the enzymes that would break down trash. And for example, acid proteases. Proteases are to break down proteins. Hydrolases can break down multiple substances. They put water in a substance and break it up. Lipases, they would break down lipids. Caffeine promotes the production of these enzymes as well. These enzymes live in the lysosome. So lysosomal activity is enhanced as well. Very intelligent outcome here. Not just that here are more trucks for you filled with the trash and they're just parked there and we're doing nothing. Shredders that would shred that are also increased. So this is the second thing they saw. And they saw these phagosomes and lysosomes co-locating near the lipids. And I have given the references to the diagrams in here. So if you are interested, you can see them. I'll show you those diagrams at the end of this talk as well. Then this was the most interesting mechanism that they showed. So let's start from the left side. Here, there are some phospholipids. This one blue dot and two little legs, red, that is a phospholipid. The blue one is phosphate and the two little red legs are lipids. Phospholipids are used up to make membranes in our cell and within the cell. The cell membrane itself is made up of phospholipids as well. And we say that the cell membrane is made up of bilayer of phospholipids. That means there's a double layer. The vesicles inside the cell, meaning the smaller units of smaller factories within the cell, are usually single-layered phospholipids. However, a phagosome is unique, especially autophagosome. Even the phagosome, general phagosome that is formed to pick up something from outside is also double-layered because you know that the cell membrane is double-layered. And a phagosome is really formed by pinching off of a cell membrane. So of course, a double-layered part will be pinched off. However, even if we make an autophagosome within the cell, we make it double layered. It's a lipid bilayer, phospholipid bilayer. To make this phagosome, autophagosome, the cell requires a few enzymes. One is ATG7 and other is ATG5. And last time we discussed with spermidine, beclin, and mTORs. mTOR need to be less to allow the autophagy to occur. ATG7 needs to be more, ATG5 needs to be more, beclin needs to be present to help build the phagosomes and to help initiate the autophagy. So here, if you see here, this little guy who's standing on top of this phagosome and building it, this is an ATG7, for example. So not only caffeine triggers autophagy, it actually helps trigger enzymes that will build phagosomes and that would build the enzymes in the lysosomes, which will then in turn do autophagy. So it brings the production of raw materials. So it's not just a, hey, do some autophagy, I'm here. My message is to do autophagy. It actually actively takes part in expressing the genes which will create the builders, which will make phagosomes and lysosomes and initiate the phagolysosome activity or autophagy. These things just make me so excited. So how do we express this? We say that caffeine induces the production of pro-autophagic proteins. And then we know what happens next. Now, how did they prove that it does it? They took some cells and they knocked out their genes for ATG5, for example. They knocked out their gene. These cells were not capable of making ATG5. Then they put caffeine on those cells. And those cells could not do autophagy. Why? Because ATG5 was not present. This proved that the lipid breakdown as a result of caffeine was through the autophagic pathway. So if you disrupt the autophagic pathway by changing the genes, then caffeine cannot do its function. Then they saw another very interesting thing. That was, you know that when lipids break down in a cell, these lipids make free fatty acids. Those free fatty acids can go to mitochondria, and mitochondria can use them for energy production, for electron transport chain substrates. Plus, of course, we know that mitochondria uses oxygen as well. It does respiration for us. Breakdown of lipids causes mitochondrial function acceleration. However, if you overload a cell with fats, so it's a different thing. Fat-loaded cell is a different cell compared to fat-not-loaded cell which has fats in it. The fat-loaded cells, that has a high accumulation of fats in it, high amount of fats, their mitochondria reduce their electron transport chain activity and reduce oxygen consumption. Their mitochondria run only enough to do functions of the cell. Their mitochondrial extra capacity to do faster ATP production or oxygen consumption or electron transport chain function is gone. So they said that mitochondrial spare capacity for respiration is gone in fat-loaded cells. Guess what caffeine does? What they observed. If you put caffeine on these cells, so now these are already fat-loaded cells. These are the cells already having fat in them. They're already in trouble. You put caffeine on them and the mitochondrial spare respiration capacity re-engages. Mitochondria, they said, did not produce more ATP, but its capacity to run the electron transport chain faster and to consume oxygen more increased. So that was a very interesting mechanism they observed. Then they saw one more thing. As I said before, that caffeine is going to help lysosomes as well. Lysosomes are, as the name says, lyso mean to break down. Somes are really just pockets. These, you can say, are the stomachs of the cell. Lysosomes have in them enzymes that I discussed before, lipases and proteases and hydrolases and others, DNases, RNases. Plus, the lysosomes like to have an acidic environment because these breakdown enzymes use acidic environment and function better. So the researchers found that giving caffeine actually caused more acidification of the intracellular vesicles, including lysosome, which enhanced the function of the lysosome and increased autophagy. And how did they find that? They observed this by saying, they said, chloroquine, hydroxychloroquine, is known to reduce acidity. That's how hydroxy or chloroquine work. They reduce the acidity of the vesicles, which then make those vesicles not operate as at the best functional capacity. And that reduces the availability and the function or performance of the vesicles for, let's say, bacteria or viruses or other things. This is how these hydroxychloroquine or chloroquine work, by reducing the acidity of the lysosomes. So what they did was, they had cells on which they put caffeine, but in the presence of chloroquine. Now, chloroquine would go and change the acid levels or reduce the acidity of lysosomes. And sure enough, caffeine was then not able to produce its desired results because it could not create enough amount of acidity, enough levels of acidity to have the lysosomes be triggered and function as needed. This also tells us that if we are taking hydroxychloroquine and caffeine, there is kind of a competition between them. And depending upon the dose, one or the other may become dominant. Then, next, they said that mice that were fed caffeine and high-fat diets, they said these mice lost hunger. They ate less, the ones that had caffeine. And they postulated, they couldn't prove it, they postulated that the mice that were on caffeine, these mice were able to do lipid oxidation through autophagy, oxidation meaning lipid breakdown, through autophagy, and were producing their own internal ATP through the lipid breakdown pathways, and they did not become hungry to go eat more nutrients. So this is their postulation. Then they also observed that in these mice, acylcarnitins were increased. Acylcarnitins are enzymes, or actually not enzymes, they are the byproducts of lipid breakdowns or beta oxidation. So that was also observed. They also observed that in general in these cells, ATP levels were increased. So we said that in a few minutes ago that mitochondrial capacity will reduce, and they did not see in the presence of caffeine any extra capacity engaged. But generally they saw that the ATP levels in the cells had increased, and they thought it may have been because there was an extra demand in the cell to produce ATP because autophagy was going on. So again, they don't have an answer to this. They have a postulation. Then, as I mentioned before, they co-administered caffeine and chloroquine, and they saw that chloroquine could inhibit caffeine's function by changing the acidity of the lysosomes. Then here, this is something I discussed in the early part of the summary as well. There were various groups of mice, and they were given foods that would increase lipid levels and that would make them gain weight and so on. And one group was given or administered caffeine in addition to such foods, and other group was not. And they saw that the group that was receiving caffeines, that group had little or no, if you see here, little or no intrahepatic lipid accumulation. In the presence of caffeine, the same food, they said that mice were eating the same way and drinking the same way. The same food in the other mice that were not treated with caffeine caused intrahepatic lipid accumulation, and not only that, the mice gained weight as well. Beautiful. And final part of the discussion, of course, this will be in your mind that, all right, so what is a dose? So once again, this is a study in vitro and in vivo mice, not in humans. And the researchers said, if we wanted to extrapolate the dosage to humans, then the dosage they used in vitro was 50 micromole or 46 micromole plasma concentration, which meant when extrapolated to humans, 4 cups of coffee, 3 or 4 cups of coffee, or 5 milligram per kilogram body weight caffeine. The experiments that they did on mice, the amount of caffeine given to mice was 30 milligram per kilogram body weight, And when you extrapolate that for humans, that comes to be 2.43 milligram per kilogram body weight, which is 2 to 3 cups of coffee for a day. So of course, it's not a tiny amount. I take 1 cup of coffee in the morning. And if I take more than one, then I become all jittery and palpitations occur and so on. So you have to talk with your doctor if you're interested in it. There are people leaving comments that, hey, I take 4 cups and 5 cups and so on. So everybody is different, but this is the dosage. So now let's just very quickly look at a few diagrams, and that would be the conclusion of this. So let's see. This diagram is interesting. Here what they did was, this is a set of cells that are caffeine treated. And in these cells, if you see here, this is DAPI. That is a marker they had introduced from outside. LC3B is the intracellular lipidation, microtubule lipidation enzymes, which are an indicator of autophagy starting or phagosome production. So here, if you see external marker plus internal production of the phagosome proteins put together, you could see that autophagosomes were being built in the presence of caffeine, but not in the presence of control, meaning in the absence of caffeine. So that is one beautiful diagram. I actually want to show you the phagosomes themselves. So in here, this diagram A is a control diagram, meaning this cell does not have caffeine. On the other hand, this cell has caffeine or is treated with caffeine. I believe this is from the mice treated with caffeine. So if you see here, this crinkly rounded things, these are autophagosomes. Their lipid membranes, you can see these are 2 membranes sitting next to each other. This is a bilayer phospholipid membrane of a phagosome. Same case is over here. And then here, same cells, caffeine treated cells. So if you see here near the fats, they are making autophagosome. This is the autophagosome, this is the fat area. And if you see here, this is the autophagosome, it's going to start eating away those fats. Same thing over here. This is actually a very good diagram as well. What they're showing here, so once again, the one above is control, the one below is caffeine. So here, the most right side diagram is interesting. I cracked up when I saw this. Here what they're showing is that autophagosomes have become co-located or have gone and become constructed near the lipids. These are lipid accumulations, and these garbage trucks and the thrashers, they are co-located next to the lipids, and they are shoveling the lipids and building the phagosomes made with those lipids and then causing lipolysis or lipid degradation or oxidation. So this is another beautiful diagram. And finally, to show you, this is another diagram with the same thing, autophagosomes. And if you go to the end of this paper, if you see at the end, they will talk about the dose here. So this is the discussion. I hope you liked it. The summary of this is that caffeine can induce autophagy, especially in the liver cells. And I showed you other studies as well, which show that caffeine-induced autophagy can actually be vascular protective. It can reduce the smooth muscle proliferation of the blood vessels, which normally occurs when the vessel is under stress or is inflamed. It also reduced the restenosis, meaning when a blood vessel, for example, cardiac vessels, when they become narrowed and then they are patented by, let's say, stents, then the restenosis can occur in some cases. And they said that caffeine helped with that. And it is known that caffeine is cardioprotective for a long time. It's not a new thing. And then we saw that with the hearing cells, over there, the researchers thought that if autophagy occurs in the hearing cells, then that may cause hearing loss. So I think that needs some more research as well. So this is the discussion for today. Stay safe, happy, and healthy, and I would see you next week.